Hi folks, today we're going to run through 4 examples of problems that you will encounter on a test. These are the key genetics problems that we're working on this year. So good examples of each of them. Some of them you've seen already, some of them will be new. But let's go ahead and get started. I think that we're going to start by doing this one. This is a monohybrid cross, which means we're just talking about one single trait. So in this case, we say purple flowers are dominant to white flowers, similar to what Mendel was doing. We want you to determine the expected phenotypic ratio if the heterozygous purple flowered plant is crossed with a plant with white flowers. Okay, so first we need to identify our symbols we're going to use. We're going to use capital P for purple, and I'm going to use lowercase p. I'm going to put a little tail on that, so these are different for white. Okay, so now we need to think about what does it mean for it to be a heterozygous purple flowered plant. Well, that means that it's going to be a big P and little p. Okay, and then the second plant, second parent in this, is a plant with white flowers. Well, if white is the recessive character or trait here, we need to make that little p. Okay, so these are the parents. We'll write this in, parents. And you always start by writing the parental genotypes. Now we need to think about what types of gametes these two parents could make. Well, we know in meiosis that only the gametes get one of the two possible chromosomes. So we're going to show these here. It could receive a big P, a chromosome with a big P, or a chromosome with a little p. These are the two possible gametes. And we could say for this plant that these are the sperm cells that are produced in the pollen. Okay? For the second one, the gametes are either going to contain a little p or a little p. Really only one possibility. Let's say these are the female eggs. Okay? What Punnett square does is it combines possible eggs and possible sperm cells to get an offspring. So we're going to get a big P, little p here. We're going to get a little p here. Big P, and little p. Okay? And then we need to identify the phenotypes of these, so the appearance. So this is white, this is purple, right? And this is purple. Okay, and so the phenotypic ratio is 1 purple to 1 white. Another way of saying this, and that's a reduced, you could say 2 to 2, but we would really prefer to have it to be a reduced ratio. We could say 50% of offspring are purple and 50% white. Okay. All right, let's move on to a second one. I lost my place here a little bit. Where did I go? Okay. Second one, a dihybrid cross. So now we're talking about 2 traits on 2 separate chromosomes. So we say purple flowers are dominant to white again, and tall are dominant to short. So let's do this. Big P equals purple, little p equals white, and big T equals tall, and little t equals short. Okay. Determine the phenotypic ratio if a heterozygous tall plant, okay, so it's tall, that means it's going to have one big T allele. It's heterozygous, so it's one of each. With white flowers, so it's going to be white flowers, it's going to be little p, is crossed with a plant that is heterozygous for both traits. So that means big T, little t, and big P, little p. Okay, so we've got that done. Now we need to think about what are the possible gametes the plants could produce. We know that a gamete is only going to contain one of the 2 alleles that you have for each, or one of the 2 one of the two alleles you have for a given trait. So it's either going to contain this little p or this little p. And those could be matched up with a little t, right? Or it could be matched up with a big T. Okay, and it could be the same for this little p and this little t and this little p and this big T. But really we get two possible gametes. We get big T and little p, and we get little t and little p. These are two possible gametes that could be produced. This individual, you could see it could be big T with big P. It could be little t with little p. So let's write these down. Big T with big P. Little t with little p, we said. And then we could do little t with big P, little t with big P, or we could do big T and little p. That's the other possible combination. Big T and little p, right? So we can see with one parent you get two possible gametes that could be produced, different genetically, and with the other you could get four possibilities. So that's why I have my 2x4 Punnett square here. So I'm going to put big T, little p. These are the possible gametes for parent number 1. And then little t, little p. And what's next? Let's do big T. I might write these down in a slightly different order. Big T, big P, big T, little p, little t, big P, and little t, little p. Okay? And then we need to go through and do our Punnett square. So in this case, we're going to get a big T, and a big P, little p. All of these cases, our offspring should have the same number of letters as what the parents did. Right, so we have in our possible offspring we have two alleles for height and two alleles for the color of the flower. This one we get big T, big P, little p. This one big T, little p, little p. Big T, little p, little p, and then big T, big P, little p, little t, big P, little p, and then big T, little p, little p, and then little t, little p, little p. Now, so we actually need to think about the phenotypic ratio. We need to go through these and identify what we have. So I'm going to say this one, this one are all the same. They all have at least one big T and one big P. So that means they're going to be tall and purple. Tall and purple. So we have that phenotype. This one are going to be tall and white. Are there any other tall and whites? Yeah, this one too, tall and white. Okay, so we have 3 of those. And then we have this one that is short and white. Right, or short and purple, sorry, short and purple. And then we have, let's give that a different symbol, short and purple, and then this one we have that's short and white. So let's count these up again. We had 3 that were tall and purple. We had 3 that were tall and white. Tall and white. And then we had 1 that was short and purple, to 1 that is short and white. Okay, could use a little more space for that, but we have a 3 to 3 to 1 to 1 ratio, and always we need to indicate the numbers in the ratio along with what the actual phenotype is. So 3 tall and purple to 3 tall and white to 1 short and purple to 1 short and white. We could also say we expect 3 out of 8 to be tall and purple. You could convert that to a percentage if you wanted to. 3 divided by 8 times 100, that gives you the percentage. 3 over 8 we would expect to be tall and white, and 1 out of 8 we'd expect to be short and purple or short and white. Okay, so let's move on to the next one. Okay, what is the expected phenotypic ratio if an individual with type A blood that's heterozygous reproduces with an individual with type B blood that's heterozygous? Now we have some symbols here. We have, this represents a type A allele, this represents a type B allele, and this represents type O. Okay, and we use these symbols because type A and B are codominant. They're both expressed in a heterozygote. Okay, and type O, this is type O, is recessive. Okay, so we show a small I for the recessive type. Okay, so when we say it's type A and it's heterozygote, that means it is this. Okay, it could not be this IA, IB, because this phenotype would be type AB blood. So it's not that. Let's erase this. And so we're talking about this crossed with an individual with type B blood that's also a heterozygote. So they have one allele for type B blood, but they also have the recessive O allele. Okay, so these are the genotypes. We put them on the Punnett square. You could get a gamete with this allele or this one as produced by parent number 1. By parent number 2, you could get this and this. Okay? Combining them, we get this and this. We get this type and we get this type. Now if we look at this, our phenotypic ratio is 1, that is AB, that's AB blood, 1 that is A, that's this individual here, 1 that is B, 1 type B, and 1 here that is type O. Okay, has the double recessive O blood. Okay, so that's our phenotypic ratio. If your parents were these types, right, you would expect 25% of the offspring to be AB, 25% to be A blood, 25% to be B blood, and 25% to be O blood. Okay. Now, the last one, color blindness, sex-linked traits. When we do sex-linked traits, we're always talking about a male crossed with a female. And we use these symbols because for sex-linked traits, the alleles we're talking about, or the genes we're talking about, are generally found on the X chromosome. So we need to show this because males don't have 2 X's, they just have 1. And so what we're going to do here is we're going to use N for normal vision and little n for color blindness. Okay, and so what we say here is determine the phenotypic ratio of the offspring if you cross a colorblind male. Well, a colorblind male is going to be this. It's going to have a little n allele for color blindness. It does not have anything on the Y chromosome because the Y chromosome isn't really homologous to the X. It's a much shorter chromosome, and you're not going to find that, you don't find the color blindness gene there. So males only have 1 gene for color blindness. It's what they inherited from their mother. Females, though, have 2 because they have 2 X chromosomes. A female could be a carrier, so she could have a normal vision allele, normal color vision allele, and one that's for color blindness. So it might look like this. Then we put these on the Punnett square, the same as we did before. You could get a gamete that has the X chromosome with the normal vision allele, normal color vision allele. You could get an X chromosome with the color blindness allele. So this is what's coming from the female. From the male, you could get an X with the color blindness allele or a Y chromosome. It doesn't have that gene at all on it. And then we combine these. Again, we get this. We get this. All right. What do all these things mean? Well, for a female, you have to have 2 color blindness alleles to be colorblind. So we have this one here that is colorblind. We have this one that's normal, but she's a carrier. We have this male who's normal, and we have this male who is colorblind. Okay, because he has one of the recessive colorblindness alleles. Our phenotypic ratio, we could say, is this: 1 normal female, 2 1 colorblind female, 2 1 normal male, 2 1 colorblind male. Okay? Other ways of talking about this, you could say 50% of females in this case, 50% of females will be colorblind. You would say 50% of males will be colorblind. You could also say 50% of females born will be normal but carriers. Okay? Now, normally females are not born with color blindness. This would be pretty rare to have a male that has the color blindness, is colorblind, and a female that's also a carrier. This allele, the little n allele, is just pretty rare in the population, so to have these 2 individuals actually find each other and have offspring doesn't happen very often. So it's not very often that you get a colorblind female, but it does happen. And I think that's it. We've gone through several examples. I hope this is helpful. You want to make sure that you understand all of these for the test, so you can go back and review these problem-solving techniques for each type, if that's helpful. Thank you.